Welcome back. This is the third lecture video on the cell. So how do things get in and out of the cell? This is going to be very important. So as I mentioned before, there are some substances that will easily cross the membrane, some substances that will require something like a transmembrane protein that forms a channel for it to be able to pass through. And then some are going to need a little bit more effort or energy in order to be able to move through them. And so we're going to look in this section at how substances cross the membrane without energy, how they cross it employing energy, and then the unique way in which water will cross the membrane and how it can affect the the shape of the cell. All right, so let's start by looking at movement across the cellular membrane. All right, so all cells are going to have something called a concentration gradient, or I should say all molecules will have a concentration gradient. And this is where whatever is located inside the cell may be in greater quantity than whatever is located outside of the cell. So let's assume for a moment that there is a cell, say a red blood cell, that is hanging out in the blood. And there is a lot of sugar outside the cell, but very little sugar inside the cell. This is a concentration gradient of sugar. One side has more, the other side has less sugar. And this will determine the rate or the ability to move in or out. Right? Diffusion will be when things can easily move across a membrane, or it's moving of molecules across the membrane. And there's going to be different ways in which those molecules can move. Simple diffusion, via simple diffusion, the molecules will move from wherever they're in greatest concentration to wherever they are in less concentration in order to equal out the amount of solutes. So you see here in this example, this could be something like, say, oxygen. So oxygen is a gas that can easily pass through the plasma membrane without requiring any additional help. So if you can imagine that there's a lot of oxygen hanging out outside of the cell, but very little oxygen inside of the cell, then oxygen will naturally flow inside the cell via the plasma membrane. Okay, this is through the concentration gradient. This is called simple diffusion. Does not require any aid, does not require energy for these to move inside the cell. Right? A key example of simple diffusion in your household would be the aromas of coffee or bacon cooking on the stove. So imagine that in the morning you wake up to the smell of bacon. How do you smell the bacon? It's not because your bed is right next to the stove. It's because if you're imagining a stove, right? And I'm not very good at drawing, but imagine there's a stove and you've got a frying pan full of bacon hanging out on this stove. There's going to be molecules coming out as the bacon is cooking, and those molecules, called aromas, are going to start to disperse through the air or diffuse through the air. Because there's a high concentration of those molecules in this area, they will naturally start to flow from high concentration to low concentration without requiring any energy. So they are flowing in favor of their concentration gradient from high concentration to low concentration and to diffuse completely through the air. And that is why you can be at a distance from the stove but still be able to smell the aromas of that bacon or coffee or whatever is being made. Right? This is the same thing happening in your cell. If there's a lot of a concentration of something that can easily pass through the plasma membrane, it will move in favor of its concentration gradient from high concentration to a low concentration. Simple diffusion. Requires no energy. There's another type of movement that is called facilitated diffusion. So in facilitated diffusion, you can imagine something that wouldn't easily pass through the plasma membrane because, say, it might be a polar molecule. So in this case, it could be something like water. Water is a polar molecule, right? So if water needed to exit or enter the cell, it could do so via facilitated diffusion because it would use some transmembrane proteins or what are called channel proteins, right? You can think of other polar molecules that might need to cross the cell. So salt would be another example of a polar molecule that would need to, hold on a second. I'm trying to erase this. I apologize. So if you remember, salt, NaCl, will be formed by an ionic bond where sodium and chloride are going to become sodium positive and chloride negative. So it forms ions. So if ions of sodium and chloride want to pass through the cell, because they are hydrophilic, they won't easily pass through the plasma membrane. Instead, they're going to need to pass through proteins embedded in the plasma membrane that are called channel proteins. So then these salt molecules, if they're in higher concentration in one area, they can easily pass through the membrane. So in this example, I'm going to make it that there's a lot of salt, a lot of salt in here, very little salt out here. So then what's going to happen is the salt will then pass through this protein in favor of its concentration gradient from where there's a lot of it inside the cell to where there's very little of it outside of the cell. This would be an example of facilitated diffusion. It's still passive transport because it's not requiring energy. And it's still molecules moving freely in favor of their concentration gradient from high concentration to low concentration, just like you had in the simple diffusion. The only difference is now they'll need assistance from a protein carrier or protein channel to allow them to pass through. So the difference between simple and facilitated diffusion is one requires assistance, one does not. Both are passive, neither one requires energy to do it. And then we're going to look at the third type of passive transport, which is called osmosis. And this is specific to water movement to be able to reduce the amount of solutes in or outside of a membrane. In this case, in or out of the cell. So in the case of osmosis, you're going to have movement of water through the plasma membrane when it needs to reduce the amount of solutes that can't easily pass through the membrane. So a good example of this is sugar. So when you have, for instance, with blood cells in the bloodstream, if you have a lot of sugar outside of the cell, but very little sugar inside the cell, then the sugar would want to naturally move into the cell in favor of its concentration gradient. However, sugar molecules are just too large to freely pass through the plasma membrane. So therefore, they're not going to easily be able to diffuse. Instead, what's going to happen is that water is going to move through the plasma membrane to try to equal out the amount of sugar that there is by doing something called, by simply reducing the amount of solute in the concentrate. So water molecules will then start to move outside of the cell. Okay. Water will move outside of the cell, and then by having more water outside, it's going to reduce the amount of sugar that is in the solution, in the watery solution. Okay. Again, this does not require energy, so it's still a type of passive transport, but it's going to be specific to the movement of water to reduce or dilute the amount of solutes inside or outside of a membrane or a cell. Okay. All right, so I think I have a picture here of this. So here you can see in this beaker, we have a high level of sugar on this side of the beaker and a low level of sugar on this side of the beaker. Because there's such little amount of sugar, the sugar is going to want to naturally move from this side to this side. However, it can't do that because this membrane that we have here, this plastic divider, is not letting it happen. Okay. If I remove the divider, it would naturally move, but with the divider in the way, it can't naturally move. So if this was an example of a membrane, where instead of a plastic divider, where the sugar can't pass through because it's just too big to fit through the membrane, then what happened is that the level of water would move. So you'd have water instead moving this way on this side. And what would happen is that with time, you're going to see the water level rise in the beaker because the water is trying to get onto this side of the membrane. Sorry. All right, let's talk about active transport for a moment, and then we're going to talk about what happens to cells with osmosis. So active transport is when you are required the use of energy to be able to move substances. And this is usually because the substances are moving from a low concentration to a high concentration, or because you're just moving too much solutes at once, right? So examples of types of active transport are also going to be three. Just like we mentioned before, three types of simple transport. Now we're mentioning three types of active transport. In the simple transport, you had simple diffusion, facilitated diffusion, and then your osmosis. In active transport, which does require energy, you're going to have carrier-mediated transport, endocytosis, and then exocytosis. In carrier-mediated transport, it's where something is trying to move against its concentration gradient. So say you had a lot of potassium inside the cell, and for some reason, and you have little potassium on the outside of the cell, and for whatever reason, you want more potassium to come into the cell. Then what happens is, in order for these potassium molecules to move into the cell, since they will be going against their concentration gradient, they are going to require energy to move them. And this is what we call active transport or carrier-mediated transport, where a protein pump will have to actively move that potassium against its concentration gradient. A key example of this is the sodium-potassium pump. Okay, sodium-potassium pump. And the sodium-potassium pump is a key example because it's something we're going to see a lot throughout 168. It's going to be heavily involved in keeping a concentration gradient around nerve cells so that they can fire off messages. And the way they're going to do that is there's going to be a concentration of sodium and potassium inside and out of the cell. And then the sodium and potassium are going to be moving against their concentration gradient to increase the level of energy available on that cell membrane to be able to fire off messages. I am not going to tell you which way it's going to move because one of the things I want you to do is be able to look at the sodium-potassium pump in more detail. And we'll be looking at that when we get into the nervous system, right? So hold off on that for now. The next one I want to talk about is endocytosis and exocytosis. And this is where you're trying to bring in large quantities of solutes basically into the cell, or substances into the cell. And because you're trying to bring in such large quantities of the substance, they can't easily pass through the membrane. They can't fit using the simple diffusion. They can't fit via facilitated diffusion either because the space is just too big for even a protein that's sitting inside the membrane. So instead, what's going to happen is when you have your plasma membrane like this, part of it can engulf a large quantity of substances with something called a vesicle to bring it into the cell. So you can imagine large quantities of substances out here that need to be engulfed. Or it can take large quantities of stuff. So for instance, if you're producing all of a sudden a lot of hormones, the blood sugar level goes up in your body because you ate too many donuts and drank it down with a large amount of coffee. Your body quickly wants to send out a hormone called insulin to tell cells in their body to get rid of excess sugar. So you're producing a lot of hormones suddenly. If you remember, if this is a nucleus, it's got an endoplasmic reticulum around it. Some of that endoplasmic reticulum will be rough, called ER, which helps to make proteins that are going to exit the cell. And the rest of it's going to be smooth, not have ribosomes attached to it. That part is going to make hormones, like lipid-based hormones. For instance, insulin. And so what's going to happen is that the insulin is going to be produced in large quantities. It's going to have to then go through the Golgi to be shipped and packaged. And then from there, it'll be sent out in a vesicle, a large quantity of those hormones in a little vesicle. And it will merge with the plasma membrane and essentially dump out its contents, dumping all those hormones out into the bloodstream. All right, and that is known as exocytosis. Understood? So endocytosis, bringing a large quantity. An example could also be a bacteria cell that a white blood cell wants to engulf in order to be able to destroy it. That would be a type of endocytosis known as phagocytosis. And exocytosis is removing a bulk amount of material from the cell. All right. So here is a look at those three types of passive transport I talked about. The first one being your simple diffusion. The second one being your carrier-mediated transport, or what's called facilitated diffusion. So simple, facilitated osmosis. They're all three types of passive transport, so no energy required. Remember that. You don't require energy to move them. And they're moving in favor of their concentration gradient. So from high to low, okay? From high to low. In the first one, it freely moves to the membrane. In the second one, it needs to pass through a channel in order to enter or exit the cell. Still not requiring energy. And in the third one, which is osmosis, it's where water is going to be moving to reduce the amount of solutes inside or out of the cell. And then you have, sorry, one moment here. Okay. I want to talk a little bit more about osmosis with regards to what happens to your cells. Okay. So let's take a little breather here for a moment. So with regards to your cells,